Hi everyone, my name is Felix Shaw. I work at the Elam Institute in Norwich, and I'm going to talk to you about some recent developments in single-cell schemas that we've been working on, and the data and metadata brokering platform COPO that I have been developing for a number of years. Elam Institute is in Norwich, and it's one of 8 biological research institutes sponsored by UKRI and BBSRC. We are co-located with the John Innes Centre, St. Laboratory and the Quadram Institute. One of our Institute strategic programs is called Cellular Genomics, which is all about single-cell data sets and exploiting such. My part of that job is to develop standards and tooling around those standards to enable sort of the fairness of such data, and for its value to be maximized. So in trying to create a standard to describe these experiments, I started with the existing work that's been done, eager not to create a competing standard. And the most relevant ones seem to be MIXIS, which is minimum information for any sequencing experiment, Darwin Core, which is a standard for describing biocuration type studies, the Darwin Tree of Life metadata standard, which we were quite heavily involved in producing, MINSCI, which stands for minimum information about single cell experiments, and FANG, which is for describing agricultural and animal studies. So these are the components, the sort of forming components of the standard. So we do this in order to work within the existing metadata landscape to enable backwards compatibility and interoperability and all of that kind of stuff. We're also trying to make this applicable to different technology stacks. So we currently support VisGen, 10x, SmartSeq, and a few others. So the way it works is we have the component standard that I just showed you with the MINSCI metadata, and then some other fields which we've added in consultation with various groups around the UK and North America. We have a website, singlecellschemas.org, where you can choose the component standard at the top, which is most appropriate to your experiment type, and then the technology stack. And then it will give you, the appropriate list of fields broken into the different parts of the experiment, along with whether they're recommended or required, and regular expressions for validating them, any sort of allowed lists of vocabularies. So as I said, we've been working with various groups. We've worked with the John Innes Centre, who we're co-located with, Roslyn, Abraham, the Biocuration Cell Atlas across Europe and America, the Ag Biodata Consortium, and the Plant Cell Atlas. So we're trying to get this standard under as many noses as we possibly can in order to, make it as inclusive and as useful to as many groups as possible. The way it's been implemented in COPO, so as I said, COPO is a metadata sort of validation and brokering type tool. So you would get your single cell schema manifest, the most appropriate for your study type, fill it out as best you can, upload it into COPO, where it undergoes a rigorous cycle of validation for this particular metadata, and then it will report any errors, and then you go back to the spreadsheet and you fix them and you upload it again, and it's this sort of cycle of validation. Eventually it will pass validation, and we'll take your, in this case, RNAseq and expression data, stage it in the COPO staging area, and that's just a single curl command from your cluster. And then it's just a one-click process to upload the RNAseq and rich metadata to ENA, where that metadata, the single cell metadata, becomes first-class metadata within that database, so it's fully searchable, fully fair, fully interoperable. And then the expression data goes to Zenodo at the moment. All accessions and metadata are available and searchable through our API and are returnable either as RO crate or JSON or CSV. It's up to you. You can choose. You can also, to an extent, choose the format in which that comes back in terms of schema. We are in collaboration with Forschungszentrum Jülich, Hannah Dorfholz and Angela Krantz there, who I found out are working on similar stuff. It was an Elixir biohackathon a few years back. And so I talked to them, and the Venn diagram is pretty much well circular for the stuff that we're doing. So we're going to be having an analogous ontology for this standard, which you can see here. And that'll be really soon. For future work, we will be very, very soon accepting, validating, and depositing image-based spatial transcriptomic data. That will initially go to Zenodo and hopefully in the future go to Bioimage Archive pending the creation of their API. So I'd like to thank my team, Aaliyah, Debbie, and everyone else. Thank you.